Topic of discussion is Enterobacteriaceae. Enterobacteriaceae is a large family of gram-negative bacteria. Escherichia, Shigella, Edwardsella, Salmonella, Citrofactor, Klebsiella, Enterobacter, Capsinia, Proteus, Yersinia, Erwinia, Pectinobacterium. These are the main species for Enterobacteriaceae family. These are all gram-negative bacterium. Certain E. coli strains can be considered as true pathogen. Salmonella, Shigella, Yersinia. Among so many species, E. coli, Salmonella, Shigella, Yersinia considered as true pathogen. Now we will discuss about that morphology. Enterobacteriaceae are facultative anaerobic organism, gram-negative, non-sporing, rod-shaped bacteria. If motile, motility by Ferritrichus flagella. Maximum except Shigella and Klebsiella, all are motile one. Motility also Ferritrichus flagella presented all over the body. Then normal inhabitant of the intestinal tract of human and animals, where it is presented intestinal tract of human and animals. Some are enteric pathogen and others are urinary or respiratory tract pathogen. Enteric pathogen, urinary and respiratory tract pathogen. Facultative anaerobic, gram-negative, non-sporing, rod-shaped motile bacterium Ferritrichus except Shigella and Klebsiella. Normally presented in intestinal tract of human and animals. Some are enteric pathogens, others are urinary or respiratory tract pathogen. This all about morphology of Enterobacteriaceae. Bacterium differ based on their biochemical reaction and antigenic structure. It have more than 150 species. Each and every species differ from based on their biochemical as well as antigenic structure. Then E. coli, Klebsiella, Enterobacter, Proteus, Citrofactor, these are the species causing opportunistic pathogen to human. Then Salmonella species, Shigella, Yersinia, some E. coli strains causes obligate act as a obligate pathogen to human. In central nervous system, it causes meningitis, causing organism Escherichia. Then Klebsiella, Enterobacter, Yersinia causes pneumonia in lower respiratory tract. Escherichia coli, Klebsiella, and Enterobacter causes sepsis. It causes bloodstream infection. Then Salmonella, Shigella, Yersinia causing diarrhea in gastrointestinal tract. Then Escherichia coli, Proteus, Klebsiella, it causes urinary tract infection. Next, mode of transmission of that Enterobacteriaceae infection through contaminated food and water. Salmonella species, Shigella, Yersinia, Escherichia coli 0157H7, that serotype, causing through contaminated food and water infection. Then through endogenous urinary tract infection, primary bacterial peritonitis, abdominal abscess. Then transfer between depleted patient from weakened patient, then insect through vector, then urinary tract infection through Escherichia coli, Klebsiella, Enterobacter, Proteus mirabilis. These are the organism causing urinary tract infection. Then pneumonia caused by Enterobacter species, Klebsiella pneumoniae, E. coli and Proteus mirabilis. Then wound infection caused by E. coli, Enterobacter, Klebsiella pneumoniae and Proteus mirabilis. Then bacteremia caused by E. coli, Enterobacter species, Klebsiella pneumoniae and Proteus mirabilis. These are the mode of infection transmitted by Enterobacteriaceae family. Then intestinal infection caused by Shigella, Salmonella, Salmonella serotype type Murium, Shigella flexneri serotype B, then E. coli 0157H7, Yersinia. These are the species causing intestinal infection. Antigenic structure of Enterobacteriaceae. Gram negative is differ from gram positive wall. This one is the gram negative. So lipopolysaccharide LPS, it also called as copolysaccharide. This is a heat stable product in the cell wall that is recognized by CD14 phagocytes. So CD14 find LPS, it allow B to be phagocytosed. LPS heat, this one is the heat stable. It has three major component. First one is um, outermost polysaccharide, that is O polysaccharide, outer polysaccharide. Second one is core polysaccharide, that's common to all Enterobacteria and Lipidae. It is actually causes that inflammatory effect. Lipidae causing inflammatory effect. It is responding increasing cytokine production, fever, and causes inflammatory effect that is associated with lipopolysaccharide LPS. So first antigenic structure, gram negative bacterium having LPS, also called as copolysaccharide. It having three major component. O polysaccharide, that one is the outer polysaccharide. Core polysaccharide, this one is common to all Enterobacteria. Last one is Lipidae. This one responds for to increasing cytokine production, fever, and inflammatory effect. This one associated with LPS. Serological identification. This one important detection of maturity of this organism. O and H antigen sudden serotype are associated with severe diseases. Sudden serotype associated with severe diseases. O polysaccharide or LPS. O polysaccharide outermost portion of LPS has strain specific antigen that are presented in each genus and species. There is a lot of cross reaction from as an antibody standpoint. Flagella are almost heat stable, which is identified easily the antibody reaction. Detection of this antigen actually provided a significant amount of clinical significance because some of that pathogenic species of bacterium associated, example 0157H7 in E. coli. Some virulence factor are shared across Enterobacteriaceae and even across gram-negative organism. So, endotoxin, actually the lipid component of LPS, it activates complement, actually activating, and it get increased cytokines, get leukocytosis, thrombocytopenia. If you have high endotoxin amount, the patient can experience DIC, disseminated intravascular coagulation fever is common. Shock, even death can occur. Lot of encapsulated organism that capsules is slimy, like a egg yolk with egg white around it. Then antigenic phase variation. O and H antigen basically differ to serotype. There was one strain of E. coli 0157H7. So virulence factor, endotoxin, capsules, antigenic phase variation, OH antigen, strain E. coli 0157H7. A type 3 secretion system or basically little bacterial syringe So the bacterium has the syringe that it hangs on. Bacterium has toxin into host cell in order to either infect host cell. So if bacteria have syringe and injecting ourselves with product that either want to cause immune response or cause them to die. Heme protein, hemoglobin, myoglobin, and other iron chelating protein like transferrin and lactoferrin. These basically allow the bacteria to steal things like iron from us, which are helpful for them to grow because these are the some organisms that record higher concentration. So overall summary of Enterobacteriaceae, large family of gram-negative rod, common features ferment glucose with acid production, then oxidase negative. Pseudomonas aeruginosa only positive, remaining things oxidase negative, then facultative anaerobic organism. Bacteria can grow presence or absence of oxygen. Then it reduced nitrate. All are motile except Shigella and Klebsiella. Then all are non-capsulated except Klebsiella. Klebsiella having capsules. Then grow on bile-containing medium. Bile-containing medium, Mechanchi agar medium. Mechanchi agar medium is the selective and differential medium. It containing some component like bile salt, crystal violet, lactose, and neutral red. What is the role of bile salt and crystal violet? Means it inhibit this growth of gram-positive bacterium. Then lactose, it two type is there. It can ferment. Lactose means it producing pink color. Not fermented means it producing colorless colony. That media color only. Then neutral red indicator of pH. Color will change into pink. Based on their lactose fermentation, Enterobacteriaceae classified into lactose fermenter and a non-lactose fermenter. Lactose fermenter Citrofactor E. coli, Enterobacter Klebsiella. Non-lactose fermenter example Salmonella, Shigella, Yersinia and Proteus. What are the selective and differential medium used for Enterobacteriaceae? Food medium we are using Mechanchi agar medium, TSI agar, triple sugar iron agar, EMP, eosine methylene blue, SS, Salmonella, Shigella agar. Then biochemical test catalyze positive, oxidase negative, nitrate reduction also occur. So these are the important things about that Enterobacteriaceae. Now we will see that notes format Enterobacteriaceae. Morphology, opportunistic pathogen, obligate pathogen, site of infections, mode of infection, Thanks for watching.